The overall aim of this procedure is to assemble an ultra-light motorized microdrive suitable for making stable extracellular recordings from behaving small animals. This is accomplished by first securely mounting the connector and motor to the chassis. The second step is to shape the shuttle to fit the chassis and attach the shuttle tubes. Next, the electrode guide tubes are installed and the recording electrodes are loaded into the microdrive. Finally, transparent covers are added to protect the electrodes and wiring from accidental damage. Once properly assembled and implanted, the microdrive described may be used to record extracellular activity over a period of up to 10 weeks.
Our design and assembly procedure have evolved over time to maximize ease of use and minimize cost such that it is more practical for and accessible to a wider array of researchers than many previously published versions.
A complete microdrive is shown here. It consists of several main components: a chassis that serves as the superstructure for the drive, a motor with a finely threaded output shaft, a threaded shuttle that carries the electrodes and provides a point of electrical connection, and an omnetics connector. Shown also are the shuttle tubes, electrodes, and electrode guide tubes.
The chassis, electrode shuttle, and electrode shuttle tubes are custom components that were designed with a 3D CAD software and were machined by a local precision machinist. All other components are commercially available.
First, use diagonal cutters to trim